Natural selection acts as a ruthless editor. As an organism reproduces, biological machinery identifies genetic variants that reduce fitness and systematically purges them. Yet, when we sequence the modern human genome, we find it saturated with genetic variants that guarantee debilitating complex diseases, conditions that severely compromise our health and lifespan. The standard medical assumption is that these diseases are simply biological failures. We categorize them as mutational load, or mismatches between our ancient biology and modern environments. Life history theory requires a different perspective. An organism has a finite pool of metabolic resources, and it must allocate those resources between maintaining its own body, somatic maintenance, and reproducing. This resource split leads to two specific genetic dynamics. The first is mutation accumulation, where late-life genetic errors are simply ignored by selection. The second, much more active dynamic is antagonistic pleiotropy. This occurs when a single gene has multiple, directly opposing effects on an organism's fitness. This forces us to ask a difficult question. Are widespread genetic diseases actually evolutionary errors, or are they the successful execution of an ancient biological trade-off? The mathematical foundation for this trade-off was formalized in a 1982 paper, published in the journal Heredity by population geneticist Michael A. Rose. Rose modeled what happens when specific alleles boost early life reproduction while simultaneously harming late life viability. He proved mathematically that genetic polymorphism, the presence of multiple variants of a gene in a population, can be actively protected. The opposing traits reach an equilibrium. Rose found that the degree of genetic dominance is critical here. When the beneficial reproductive effect is dominant and the deleterious health effect is recessive, the population maintains the disease-causing allele indefinitely, even if the eventual viability costs are severe. For decades, these mathematical models were strictly tested in short-lived model organisms. Geneticists relied on massive breeding experiments with fruit flies to observe these trade-offs. While the math cleanly predicted outcomes in controlled fly populations, proving that antagonistic pleiotropy was responsible for complex, highly confounded human diseases remained theoretical. To definitively test if Rose's postulate applies to Homo sapiens, researchers needed a volume of genetic data that did not exist in the 20th century. That threshold was crossed with the advent of massive genomic databases like the UK Biobank and modern high-throughput sequencing, allowing researchers to analyze the DNA of hundreds of thousands of individuals simultaneously. In the 2026 study published in Nature Ecology and Evolution, a team led by Eva Brigos-Barril utilized this data to map the genetic architecture of 62 complex human diseases, comparing them directly against two life history traits, longevity and fertility. This scatterplot shows the genetic correlations. First, looking at lifespan. Of the diseases that showed a significant genetic correlation with longevity, 85% exhibited a negative correlation. Genetic predisposition to these diseases shares the exact same architecture as a shorter lifespan. But when the researchers cross-referenced those exact same disease risk alleles with reproductive success, the data inverted. 87% of diseases with significant genetic correlations to fertility showed a positive correlation. This overlap is highly concentrated in psychiatric conditions like ADHD, as well as across the cardiovascular system. The alleles driving risk in these domains heavily overlap with increased reproductive output. Because this data is derived directly from genome-wide association studies, it bypasses environmental and socioeconomic variables. We are looking at the shared genetic architecture built directly into the DMA sequence. The genetic factors that actively promote human reproductive success simultaneously and predictably increase human disease risk. The research team then isolated exactly where this trade-off occurs. They identified 1,142 specific independent single nucleotide polymorphisms, or SNPs, that directly link disease risk to these life history traits. They categorize these variants into two groups: positive pleiotropy, where the disease risk allele increases fitness traits like fertility, and negative pleiotropy, where the risk allele reduces those traits. To see if evolution was actively responding to these traits, they applied specific selection metrics. The singleton density score and the number of segregating sites by length. These track recent positive selection in the human genome over the last 2,000 to 50,000 years. This figure shows exactly how selective pressures have acted on these specific disease risk alleles.
Look at the blue dashed lines. These represent the alleles that increase fertility.
Across every age-at-onset category, they remain elevated above the baseline, indicating continuous positive selection. Now look at the red lines, the alleles that decrease fertility. They are completely suppressed below the zero axis. The box plots confirm the statistical significance of this gap. Natural selection is actively pushing fertility-increasing alleles to higher frequencies in the human population. And remember, these highly selected fertility-boosting alleles are the exact same variants that guarantee susceptibility to complex human diseases. Natural selection is not failing to purge these variants. It is deliberately protecting disease risk alleles because the reproductive payout is too high to ignore. At this point, a critic might point out that genetic correlation and selection signatures do not inherently prove a causal relationship. A shared genetic variant doesn't necessarily mean the disease itself causes a change in lifespan. To prove directionality, the researchers utilized Mendelian randomization. This analytical method uses genetic variants as instrumental variables to isolate directional causality, stripping away environmental noise and lifestyle factors. This circular plot visualizes the Mendelian randomization analysis. Looking at the outermost ring, which maps the causal effect of disease liability on longevity, 22 out of 62 diseases feature solid RAD markers. These indicate a statistically significant direct causal reduction in lifespan. The cardiovascular domain is particularly striking. Hypertension, coronary artery disease, and heart failure exert a massive, causally detrimental effect on human lifespan. Historically, data on longevity is heavily skewed by socioeconomic status. Variables like income and educational attainment often dictate health outcomes. But the red triangles and squares plotted here show that these causal estimates hold firm, even after running a multivariable adjustment for those exact socioeconomic factors. The data also contains weaker but suggestive reverse measurements, showing that genetic liability for certain traits, like ADHD, causally increases fertility, validating that this evolutionary trade-off operates in both directions. Genetic liability to disease strictly and causally degrades human longevity. The persistence of these alleles across generations is empirical proof of Michael Rose's 1982 mathematical postulate on antagonistic pleiotropy. To understand how this genetic architecture manifests in living populations, we have to look at demography. The critical variable is the exact age at which a disease strikes. For early onset diseases, there is a clear reproductive advantage for individuals carrying a high genetic risk, but remain subclinical or undiagnosed during their reproductive years. They reap the genetic benefits without paying the immediate physiological cost. Late onset diseases show the inverse. Because these conditions strike after reproduction is largely complete, individuals carrying the risk alleles show higher lifetime fertility, even if they are eventually diagnosed. This dynamic is governed by an evolutionary concept known as the selection shadow. Natural selection intensely optimizes the reproductive window of a human lifespan, typically between ages 15 and 45. Once that window closes, a shadow falls over the timeline. Because late-onset diseases manifest entirely within this dark zone, selection is blind to them. They escape any evolutionary penalty because the organism has already passed on its genes. This age-sensitive trade-off allows carriers to harvest the fertility-enhancing benefits of risk alleles in their youth, effectively delaying the biological bill until reproduction is secure. Evolution ignores your late-life morbidity, precisely because it has already secured your early-life fertility. In 1982, Michael Rose calculated the mathematical threshold where the viability cost of a gene is mathematically outweighed by its fecundity advantage. Today, the UK Biobank and massive genome-wide association studies have delivered the molecular, population-scale proof that this equation runs continuously within the human genome. This reframes how we view medicine. Senescence and susceptibility to complex diseases are not biological glitches, nor are they modern anomalies. These diseases are baked directly into our species architecture. They are the calculated, unavoidable cost of our reproductive success. For modern medicine, this means targeted interventions and personalized therapies must account for deep evolutionary constraints. Curing a genetic defect might disrupt an ancient balance. We don't get sick because natural selection failed us. We get sick precisely because it succeeded.